Welcome to the GEOTOP-A seminar. Today we have Christine Heitsch from Georgia Tech, who's going to talk about barrier height and other problems in RNA branching landscapes. Take it away, Christine.
DeWitt, thank you so much for the invitation, and thank you to you and all of the organizers for this opportunity. I'm excited to tell you about some of these results. But first, I really have to thank DeWitt for the profound impact that he had on my career. I was not privileged enough to work with him, but I benefited from attending meetings that he organized in Santa Fe as part of the program for mathematics and molecular biology, and that program and the genuine interaction that it fostered between math and bio was really a key inspiration in the Southeast Center for Mathematics and Biology. And so I hope that he appreciates just the profound impact that he has had on mathematical biology.
Well, that's very kind. Thank you.
So as you will know, I'm not really a geometer nor a topologist. What I work on is I would describe as discrete mathematical biology. So I am primarily interested in the interaction of combinatorics with molecular biology, and I really am most excited when it goes both directions. That is, not only do we have applications of the mathematics to the biology and generating new insights into it, but that thinking about these biological problems really should challenge us to develop a new and interesting mathematics that is important both for its application, but also just intrinsically in terms of its mathematics. So on the right-hand side, we have here, these are actually what are known as RNA secondary structures, and I'll describe that a little bit more in a minute, but they're drawn in a they're bacteria that fluoresce on a Petri dish. And so this is an example of, indicates an example of biology, because biologists primarily demonstrate they've understood a system by formulating an experimental hypothesis and then testing it. On the other hand, over here are sequences of numbers. And so if there are more combinatorial people watching this, they might recognize the first one, which is a very well-known sequence. The second one is less well-known, and its actual closed form enumeration is still open. And so we will come across both of these sequences in the course of the talk. But a combinatorial person will often demonstrate that she's understood a problem by counting something. So if I've really understood the structure of the problem, I should be able to say how many objects there are as a parameter n varies for it. To make the connection between these two a little more precise, really what I think about mostly are strings, trees, and RNA folding. So on the right-hand side, although on my screen it's not showing up so well, I promise there will be a better picture later on, is what is known as an RNA secondary structure. And it is an RNA sequence that has formed intramolecular base pairs. And on the left-hand side is, in some sense, it's graphical skeleton. So what I've done here is wherever I had these open regions, which are called loops, I've just collapsed them down to a point in the plane. I'm keeping track of where the ends of the sequence are, and that is going to be the root of my tree. And because this tree is rooted at the bottom, unlike the mathematical convention, I'm indicating that by the open vertex here. And then the stacked base pairs that connect these open regions become the edges that connect these vertices. So in a very natural way, we can move between the combinatorial object on the left and its RNA instantiation on the right. So once upon a time, one asked, why do we care about RNA? In case you are still wondering, I have to think you don't remember the global pandemic that we all lived through. So by now, most people know that many, if not most viruses are coded in RNA. That includes not only COVID-19, but also Ebola, as well as things that are less likely to kill you, but not impossible, such as the common cold and influenza. RNA is also critical in terms of its ability to regulate genes. And then there is just the very standard role that RNA plays in the in the process of converting the information that's encoded in DNA into proteins. So as a biomolecule, RNA is of paramount importance, although once upon a time it was often seen as, I will say, the redheaded stepchild to DNA and proteins. All right. So the problem that we're basically going to consider is how are branched structures encoded in these sequences? So we can think about RNA structure at 3 different levels according to the dimension in which we're thinking of it. So an RNA molecule, of course, lives in 3 dimensions, and the overall molecular conformation is essential to its ability to perform its function. So this is the 3-dimensional structure of what is known as a transfer or tRNA molecule, and it is one of the 3 RNA molecules that are involved in reading the information encoded in DNA and translating it into proteins. And this L-shaped is critical to that because the anticodon is here at the bottom and the amino acid is attached there, and it slots into the ribosomal machinery in order to perform that. So, but understanding the 3-dimensional structure of RNAs is still very much an open problem, and AI is not putting us out of business anytime soon. There just isn't enough data. The molecules are they don't crystallize very well, that we don't have a lot of data, and the data that we do have is primarily the same type of molecules. So there's a great deal of redundancy in the data that is available. So understanding the 3-dimensional structure of RNA is still very challenging. On the other hand, the 1-dimensional structure, which is just the oriented sequence of nucleotides, A, C, G, and U for RNA rather than T, is a completely solved problem now. So for any molecule, it is very easy to get the sequence information. And then the intermediate structure between these two is what is called a secondary structure. And if you know anything about proteins, you may be thinking, oh, proteins also have a secondary structure, but this is completely different. So the secondary structure of an RNA molecule is the set of intrasequence base pairs. So what happens is that this sequence here on the left, the first G actually ends up pairing with the third from last C here. And so that is this base pair right here, this GC base pair right here. And then subsequent pairs can continue to form. And so we get this nice laddering of stacked base pairs, exactly like a DNA, like a little snippet of a DNA double-stranded helix. But then it breaks down. And so it can't continue to form base pairs, but then, eventually it reaches another point where it can form another run of 4 base pairs and a run of 5 base pairs and so on. And so this is what is known as a secondary structure, is just a set of intrasequence base pairs for an RNA molecule. Now, there is, so as you may already be thinking, why, is this the only secondary structure? And the answer is definitely not. An RNA sequence has exponentially many possible secondary structures in terms of the sequence length. And so why this one? And one of the ways that we think about RNA folding is that it should basically, it should adhere to basic biophysical principles, which is it should be thermodynamically favorable. And so the expectation is that an RNA secondary structure is trying to maximize the number of stacked base pairs that form these helices. So that is a negative free energy. So it is a positive, it's a stabilizing interaction, whereas it's trying to avoid the cost of loops, and those are the single-stranded regions because those are basically viewed as destabilizing. So we have this fundamental sort of, balancing act of as many base pairs as possible, but don't have the loops cost too much. So given that very brief overview, now we're going to start talking about, so what kind of questions might you think about? Well, and a possible question, and the one that I actually first, that was my introduction to this field overall, is suppose you wanted to design an RNA secondary structure. So here is a four-arm structure, a cloverleaf, it is often called, that we've been looked at several times now. And I can take that skeleton from, I can sort of draw its graphical skeleton, and I can say, this is my target. I want a secondary structure that has this basic shape. And I'm even keeping track of the length of the helices in this case. And so that it is very easy to write down a sequence that can, conditional, fold to that secondary structure. Because what I do is I just put GC pairs along the edges where I want base pairing, and I put runs of A's where I don't want base pairing, because an A does not interact with either G or C. So I say, this is excellent. I've produced a sequence that will fold, I've produced a sequence that meets all the constraints. Except that the problem is that just because a sequence can fold to the target structure does not mean that that will be the minimum free energy one. And so what happens to this particular sequence is that it exploits symmetries between the second and the third arms, and those actually fold in this alternate conformation, which has then, which has a lower free energy than the target one. So we have this problem of not only do we have to have satisfy positive constraints to solve the design problem, but we also have a very large number of negative constraints that have to be satisfied as well, so that we don't have alternate structures that end up being better for us. And so our solution at the time was a very standard one in computer science, which was the department that I was in as a postdoc. And that is just to take the problem, this problem of designing an RNA structure, and say, we're going to reduce this to the DNA word design problem. And so we're going to say, if you could come up, we will tell you how good a word set you need to come up with. That is, we will tell you how different the encodings on the helices need to be so that the target structure is the one that you end up with. Now, for me, this actually spawned two different research directions that I have continued to pursue over the years. And so I'd like to tell you a little bit about one of them at the very end, and a lot about the other one. And so the little bit is this idea of, well, what about these loop energies? And so this has been a long-running collaboration with a former postdoc of mine, Svetlana Plesnanovic, who is now a full professor at Clemson University just up the road. And we have about 6 papers and counting, with 10 different junior researcher co-authors over the years. And so often I have given talks about that, where I just describe this whole RNA polytope project for loop energy combinatorics. But today, I'd like to tell you about something that ends up being a little bit more, well, involving a little less biology background, and so therefore ends up being a bit more mathematically accessible as well. And so we're also going to think about this helix exchange combinatorics problem. So in the design example that I gave you, the sequence exploited some symmetries to find a lower energy conformation. And we can think about, well, what if we give the sequence sort of the most scope for exploiting symmetries? What kind of structures would we get, and what would the interactions between those structures be? And so here I've got an RNA sequence that, this is the RNA equivalent of a spherical cow. So not natural in any way, shape, or form. It is just a run of 4 A's, a run of 6 C's, followed by 4 A's, sorry, G's, 6 C's, copied some number of times. In this example, copied 3 times, and then 4 A's. So you can think about the runs of A's as being spacers between the helical encodings. And so then I can ask, well, so first of all, how many structures are there that have the maximum number of base pairs? And in this particular case, I'm going to get 5 structures that all have 18 base pairs in them. And then I can ask, and how do they relate to each other? So for instance, in this structure here, the first G is paired with the first run of G's is paired with the first run of C's. So 1 is paired with 2. But then the next G's waits for the last run of C's. So I can think of 6 as being paired, 3 with being paired as 6, and 4 with 5. So here, the numbering is I have labeled each run, I've labeled the runs of C's and G's with the integers from 1 up to 6, and then I'm writing an arc between them when they have formed a helix in the secondary structure that I'm considering. And so this one interconverts with this one by keeping 1 and 2 fixed, but switching the pairing between 3 and 4 and 5 and 6. And I can move from this one to this one by keeping 3, 4 fixed, but switching the pairing between 1, 2, and 5, 6. And similarly, I have all of these reversible operations. So I can move among these 5 structures in this breaking apart and repairing way. But note that I cannot get from this one to this one direct in only one move. It takes me 2 of these breaking apart and repairing operations. But I can either go through this one where all 3 helices are incident on the central loop, or I can go through this one where all 3 helices are incident on what we would call the external loop. And so these non-crossing perfect matchings are in direct equivalence with the plane tree, the skeleton that we talked about already, and they're just a more succinct way of keeping track of this operation. It will also be interesting, and we'll come back to it, to think about, well, this one that I mentioned, this one also is not directly connected with that one. And when I take its arc representation and I take this one and flip it from the top to the bottom and move it to be there, I end up with a single closed loop. And so this is an object that is known as a meander. It comes up in all sorts of different mathematical contexts, including both topology and geometry. And then we will end up getting some new insights into meanders based on this operation that we just talked about. So let's be a bit more precise about what the objects that we're working with. So the trees that we are thinking about are what Stanley calls a plane tree. So it's a rooted tree whose subtrees are linearly ordered. If I'm thinking about trees with 3 edges and therefore 4 vertices, if they're unrooted and unordered, there's only 2 of them. If I have to choose a root, then that differentiates these 2 versions of the star tree from each other. And it differentiates the straight line from when I choose the root to be 1 away from a leaf. It is the ordering on the subtrees that differentiates this tree from that one. Because if I stand at the root here, I know that my left child is not a leaf, but my right child is, whereas the left and right are reversed here. And so the ordering is on the children is exactly what allows us to keep track of that. So these plane trees are one of the very many combinatorial objects that are enumerated by what is called the Catalan numbers. And so that is the first sequence of numbers that I had on the second page. And so they are, I think, Stanley was keeping track, had a sort of an atlas of the different instantiations of the Catalan numbers. And I think it was up to approaching 500 the last time I checked. And so they are just sort of ubiquitous across combinatorics and its interactions with other areas of mathematics. So we're going to do something a little bit different with them because we are thinking of these trees as actually really representing RNA secondary structures. So I'm going to label the boundary of the tree with a pre-order walk. And so this is exactly the pairings that we were talking about just two slides ago. So this tree has a pairing between 1 and 6, 2 and 3, and 4 and 5. And I'm going to use those pairings to keep track of the edges. So I think of each edge as basically being composed of 2 half edges, and the half edges are all labeled, and I denote an edge by its half edge labeling. We're also going to need the idea of the parity of an edge. And so we'll call an edge odd if the first index is, and then clearly it's even otherwise. And so we can see that the edges that are incident on the root are all odd edges. And then, so this is an odd edge, and then its child is an even edge, and then its child, which is the root's grandchild, is an odd edge again. So the parity is going to flip as we move away from the root vertex. And we're going to use this notation. C of t is just going to be the number of odd edges. So this tree has 1 odd edge, these 3 have 2, and this one has 3. And then we're going to, then this pairing exchange operation that we were thinking about, we can be precise. So if I have 2 edges that are incident on the same vertex, then they are composed of 4 half edges. And my pairing exchange operation is just breaking these 2 adjacent edges apart into their 4 half edges and then repairing them in the opposite way. Now, when I'm only thinking about the edges, this seems very simple, but what you have to recall is that there may be subtrees hanging off of this. So these this operation preserves the half edges and it preserves the number of edges, but it is completely changing what the biologists call the topology of a tree, and which always makes me give a little twitch. But the conformation of the tree ends up looking very different because there can be subtrees hanging off of each of these sort of parts of the vertex. And so if there were subtrees to the left and right, they were adjacent to each other and on the 2 edges on the left, and then they end up being pushed away on the right, whereas ones that were separated on the left end up being pushed together. So you've really got this flip-flop that's going on. And as a consequence, this operation changes the number of odd edges by exactly 1. So in terms of the parity. Now, this is the graph that we were looking at before in terms of the secondary structures, but I've just drawn it now with the trees rooted at the top and the dash. So the vertices of this graph are plane trees, and the edges represented by the dashed lines are these matching exchange operations. And we're going to show that this graph is always connected. And it's fairly simple to see that because for any tree, I'm going to show that there is a path that takes it to the tree that is to this tree, that is where all of the edges are incident on the root. And it's just an inductive proof, which is that if my root has more than 1 child, then inductively, this subtree is connected to the corresponding one on 2 edges, which is exactly what I end up with. If my root has 1 child, then I'm going to operate on the root and its first child, and I'm going to move to one that has 1 different. move one step closer to the tree. So for this tree, one step gets me to where I want to go. For this one, the step that I would take would actually be this one interconverting with that one. And so that takes me to this tree, which is, I already know, which is one step closer to the root. And so that my graph is connected, which tells me that it is possible to interconvert between any of these RNA secondary structures. But the path that I've just described, we would say is high barrier in general. Now, what do I mean by that? So there are a set of different problems that are known as barrier height problems for RNA folding. And really, this landscape picture is intended to be very suggestive. So barrier height problems are asking about the cost of transitioning between different configurations. So you might think I'm in this, I'm thinking about a structure in this minimum, and I'm asking, can it get to that minimum? And the answer is not particularly easily, because it would have to go over this sort of mountain range in order to get there. But if I'm thinking about, this minima or this minimum and that minimum, then it actually has a low barrier, fairly low barrier path for transitioning between them. Now, there are many different variations on this theme, and so what matters, in order to talk sensibly about a particular problem, you have to be more specific about how are you measuring the cost, what are you thinking your transition steps will be, and what exactly is the configuration space that you're operating on. So we're going to be thinking about branching as a thermodynamic barrier. And the reason for that is because we can take the free energy model that biologists use all the time to try and think about the relative stability of these RNA molecules. And we can write down how we would score one of these plane trees in a way that comes directly from this thermodynamic model. Now, what's important about this is that what is known as a hairpin, and so those are the leaves in the trees, are energetically the most expensive type of loops. A internal or non-branching loop is middling. It's costly, but not nearly as costly as the hairpins are. And then what are known as junctions, that is a loop where that has two or more children going away from the root vertex, have this particular form. And what is uh, fascinating is that this function is actually increasingly negative with the degree of the loop. And so this is telling us that the thermodynamic model is preferencing high degree loops, at least as far as like this, this analysis goes. And that is very counterintuitive because we don't see really high degree loops in RNA secondary structure. And it's just a biophysical packing thing. They actually exist, and they would have to, I can't have sort of 30 different helices all in, one clump around each other. So one says, is the model just completely wrong? And the answer is, and can be shown mathematically, that it's not, because the fact that branching is locally favorable is actually globally balanced by the cost of these hairpin structures. And so in the tree model, we can assign to the branching, so every branching loop produces it sort of, you think about it as kind of a flow argument. So the root starts with having one hairpin to it. And then as I move through the tree, if I don't branch, I just pass on the hairpin to my child. If I branch, then I have to spawn I can pass on one hairpin that I inherited from my parent, but then I have to spawn, then every other child also needs a hairpin. And so I have to spawn more hairpins to pass on to them. So I am increasing the number of hairpins in the structure every time that I branch. And that means that you can write down a function and calculate the optimum to show that the the minimum energy is going to be obtained where you branch as much as possible. So you pick up the benefit for branching, but you keep the degree of branching as low as possible. And so that is going to have the maximum number of vertices with degree 2. So if we go back to our graph, then the problem here is that the paths are all taking us through these very high branching degree trees. So, I proved to you that every tree has a path to any two trees have a path, are connected by a path, but that path has to go through this tree, which would be prohibitive then if we're thinking about it in terms of barriers. And so we're going to think about this characteristic of branching skew here. And branching skew is defined as the number of odd edges minus n minus 1 over 2, because it's actually going to be symmetric around the middle of the graph. So the two extremes have the highest amount of branching skew, and then the middle one, if it is odd, or the middle two, if n is even, are going to have the lowest degree of branching skew. So we're going to so our configuration space is this set of plane trees with n edges connected by these pairing exchange moves. So our steps are one of these pairing exchange moves. And then this is our cost function, is this branching skew which approximates the loop energies. And then we're going to ask about if we can characterize minimal skew paths that first of all move only left to right. And so we'll call those forward paths. Next, we'll think about when they reverse direction exactly once, and we'll call those V paths. And then we will recognize that those are fairly artificial constraints. And so we'll think about, how much or how close can we stay to the branching skew that we started with? And so those are going to be one away from optimal. They're either bounded or tightly bounded. And then we'll also start considering the length of the path as well, and to see when how often are the bounded or tightly bounded ones actually geodesics. So the key insight is to understand this loop decomposition of the trees. So I've not tried to write down the definition because it's quite technical. We're just going to go through an example. So I want to figure out how T relates to T prime. Okay? And so T has its arc diagram will be just the rainbow one. And so here T is on the top. And I'm going to consider how it relates to the two star trees first. So this is the star tree where it has only 3 vertices of 3 leaves, and this is the star tree where it has all 4 incident on the root. And so the rainbow arcs are on top, and then I have for this tree U4, I've just got one arc from 1 to 2, 3 to 4, 5 to 6, and 7 to 8. And so I get 2 different closed loops here. And those are my simply aligned subsets. So the way that I get these simply aligned subsets is I take one tree that I'm interested in, I take the other tree that I'm interested in, I look at their non-crossing perfect matchings, that is their corresponding arc diagrams, and I put one on the top and one on the bottom and trace out the closed loops. And I'm going to see that I get, and I'm only going to pay attention to the odd edges here. And so here, this gives me a partition of this particular one gives me a partition of the odd integers that consists of 1 and 7 and 3 and 5. And if instead I do it for this other star tree, I get a different partition, I get now 1 is by itself as a singleton, 3 and 7 belong to the same closed loop, and 5 is by itself. And so I've gotten 2 partitions of the odd integers according to these trees. Now, if I consider t and t prime, I have that 1 and 5 are in the same loop, and 3 and 7 are in the same loop. But the problem is that when I go back to the trees, those closed loops don't correspond to subtrees in my tree. So with the star trees, this did not pose a constraint because the edges involving 1 and 7 were a subtree in this one, and 3 and 5 were a subtree. For the second one, for this one, 1 was a subtree, 3 and 7 were a subtree, and 5 was. But now, if I look at 1 and 5, they are not a subtree. And so this is a problem. But it is 1 and 5 are a subtree here. If I look at 3 and 7, 3 and 7 are a subtree here, but not here. So what I need to do is to take the union, I need to take the minimal union of closed loops so that I get subtrees and rather than just disjoint subsets. And so the partition that I get of the odd integers, according to these 2 trees, is actually the whole set itself. So once we have this idea of partitioning according to the trees, then that actually tells us allows us to exactly characterize the forward paths. So I'm going to say that T has a min-max decomposition with T prime, meaning there is a forward path, and I'm going to claim that's equivalent to T having a forward path to T prime. Exactly when there is a tree partition for those 2, where the induced subtrees are each a star tree. And then the choice of the root depends on the edge parity. And so the fact that this min-max decomposition is equivalent to a forward path, in one direction, it's easy because if I'm thinking about just operating on disjoint subtrees, then I can do pairing exchanges on them are independent. So that allows me to move freely and not disturb anything else. In the other direction, I'm going to inductively construct the partition that I need. And this works exactly because of keeping track of the parity. And this also then allows us to construct the apex of a V path, because if I given 2 trees, then the apex is going to be, I take their tree partition and I patch it together from the correct parity star trees. So that's, we've done 2 out of our 4 path questions, but what we've actually done in some sense is reinvented the wheel. Because if I look at this graph, I can prove lots of other things about this graph besides the fact that it's connected. I can prove that it's n-partite, meaning it comes in these ranks, that it has a diameter of n-1, and the ranks of those disjoint sets are enumerated by the Ariana numbers, which will come as no surprise to know that they're a well-known decomposition of the Catalan numbers. So what we've actually done is we've basically rederived many of the properties of what is known as the lattice of non-crossing partitions. And so this graph that we have can be shown to be isomorphic to the Hasse diagram of that lattice. And so, sorry, there's a fan that just started up behind me, and I was like, and so that's and so we ask ourselves, okay, so now, did we learn anything more by doing it in this tree way rather than in the standard way? And the answer is actually yes, we learned quite a bit more. So if you're not familiar with a non-crossing partition, it is basically, it's a partition of the integers from 1 up to n, which when written on a circle, the parts of the partition do not cross each other. So this is an example of a non-crossing partition. That is an example of a non-crossing partition, but 1, 3, 2, 4 would be a crossing partition. And so that is not included in the lattice of non-crossing partitions. And so we have this, we have a way of mapping trees to non-crossing partitions, but it actually allows us to then characterize an operator on the lattice of non-crossing partitions whose structure had not been understood. So this is called the Crevaross complementation operator, and it was challenging for people to count it because it actually, the standard definition that Crevaross originally developed in 1972 involves an interleaving between two different partitions. And so that made it difficult to keep track of trying to enumerate how many orbits are there under this operator, because it's not an involution, and what are the size and structure of the orbits of this operator. But if we think about it in the tree, in our tree version of then actually we realize all we're doing is rerouting the tree. So every time we, in this Crevaross complementation operator is exactly just changing the order, changing where the root is on the tree by 1 index. And so that then means that the orbits can be counted by leveraging either standard methods or the cyclic sieving phenomenon that Reiner, Stanton, and White developed. It also then leads naturally to this meander problem that I mentioned, which is, we're thinking about these pairs of non-crossing perfect matchings, and meander is exactly one of those pairs that it generates only a single closed loop. And so one of the ways and that corresponds to, those pairs correspond to plane trees that are diameter achieving. So anytime I have two trees whose distance in the graph is exactly n minus 1, then I know that they are going to give me a meander. And so we could think about, well, how do these meanders relate to each other? So in this instance, we've got here, I'm fixing the tree, I'm fixing the set of arcs that's on the bottom, and I'm thinking about all possible arcs on the top that would generate a meander. And they are related and under pairs of these matching exchange operations. And so I get these, when n equals 4, I get these 4 different graphs, a triangle, a square, and a singleton. When I move to n equals 5, I get 5 different graphs, and they have some quite interesting symmetries to them. So it's not surprising that the rainbow arcs have a left-right symmetry, because of course the rainbow itself has a left-right symmetry. And it's not surprising that this one has also has a left-right symmetry. It's not clear exactly why it has a top-down, top-bottom symmetry, and it's really not clear why it has a permutation symmetry of order 3, for instance. So these sort of meander graphs are provably connected, but very little else is known and might provide additional information, both in terms of the maximally different RNA branching configurations, as well as this long-standing open problem of trying to enumerate meanders. So we've wandered pretty far away from RNA secondary structures. So we'll kind of start making our way back. And part of that is going to be thinking about these zigzag paths. So these are ones that are going to be one away from, at most one away from optimal. And so the idea is I'm allowed to sort of, I'm allowed to literally go back and forth between trees. So, this one is uh, this one and that one are not directly connected, but maybe I go back and then I go forward and then I go like this kind of thing. And so I can bound the branching skew tightly. So I can zigzag back and forth and get between the two, but the length of that path grows quadratically. Whereas the geodesic has at most k minus 1 steps, because where a k is just the number of simply aligned subsets. So basically, I can essentially move I can move between the two trees by moving on the aligned subsets in a disjoint way. And the geodesic is tight. So I have a bounded geodesic, which is tight unless my trees start off with the same number of odd edges, exactly when, or if they have a block decomposition, meaning that the induced subtrees are also simply aligned, because in that case, then they don't interact with each other in any particular way. But it would be of interest to have, to be able to bound the length on the zigzag paths more tightly. This is a very rough upper bound, and also to know under what, if this is a block decomposition is sufficient, but it's not obvious that it is also necessary, and so that's another question that we don't know the answer to yet. So I claim that these relate to RNA hairpin stability and domain architecture, and that is because the geodesic actually the minimal paths are because you first you find a hairpin that exists in your target tree, that exists in T prime, but not in T. And then those edges, you're guaranteed to be able to operate on them. And so you fix the hairpin. And then you can sort of work your way out from the hairpins, fixing the edges as you go, so that you can move between the trees. And so that's how you get the geodesic has n minus k steps. And that the domain architecture is that this block decomposition is exactly our ability to decompose the trees into subtrees that don't interact with each other, which is essentially what RNA domains are defined to be. And so there are there may be possible bio applications down the road. It's definitely the case that RNA misfolding for hairpins is implicated in its role in Huntington's disease. And so this is a picture I pulled from that paper where they're showing that the hairpin, this misfolded hairpin, this hairpin that shouldn't be there actually recruits proteins that then start forming these problematic plaques. And then this is an example of a domain refolding where they identified a motif in the RNA virus structure that affects the protein expression and seems to mediate the infection. So, it may be that there are useful biological insights to be gained from this. But I want to, before wrapping up today, I want to convince you that I do really genuinely care about the biological applications. It's just what I've told you, what I've spent most of the time telling you about, the potential applications still are in the process of being developed. But I have a couple of slides to tell you about this RNA polytope project, because that is a much better, a much longer running project, and so better developed. And so we have spent a lot of time and energy trying to improve thermodynamic predictions by thinking about the branching of them. And so this is an example. We're back to thinking about our tRNA sequences. And so these are two possible secondary structures for a particular tRNA sequence. It's supposed to, if it's going to be a functional tRNA, form the cloverleaf structure on the right. But the nearest neighbor thermodynamic model thinks that the three-arm structure, sorry, on the cloverleaf structure on the left, but the nearest neighbor thermodynamic model thinks the three-arm structure on the right is more energetically favorable, but not by a huge amount, just by a relatively small amount. But tRNAs are one of the most, essential RNA molecules. It's the one whose structure is best understood. And so a very reasonable question would be, how is it that the thermodynamic optimization, when it predicts a minimum free energy structure, has a typical accuracy that's only about 50? And so meaning that, you can have structures that are perfectly predicted, where every base pair is perfectly predicted. You can have ones where no base pair is predicted correctly, and just about every much that is in between. So, if you were to ask this question to a biologist, they would most likely say, "Oh, well, tRNAs, they're very special, they're different, and there's so much that the thermodynamic model doesn't, so many factors that the thermodynamic model doesn't include. So, it's not reasonable to expect the model to do particularly well for tRNA." And Svetlana and I, because we've had this running argument with reviewers all the time, would say, yes, tRNA is special, and there are many, many factors that the optimization doesn't include, but we can show you that the branching parameters also matter. So, as I mentioned, Svetlana was a postdoc with me. This is a picture from that era, but she's now a full professor in the Clemson math department. And so we have this whole long-running project where we're using techniques from geometric combinatorics to give a parametric analysis of the RNA branching. And so the basic idea encapsulated in this picture from Reka Thomas's lectures in geometric combinatorics is that we're thinking about a linear program. And so that's this line, and we're optimizing over a convex set, and that's the square. Now, our convex set is actually the set of all possible secondary structures, more or less. And so it's a four-dimensional space. And our line is actually a four-dimensional hyperplane. But really, what we're most interested in is the complexity, is the trade-off in the branching entropy function. And so we're going to use this duality between the convex polytope and the normal fan that allows us to fully characterize those parameters. And in recent results, we've been able to show that it's really that the math has a profound effect on our ability to accurately predict tRNA structure. So this particular picture, the blue trapezoids represent the best set of parameters for tRNA, and they are both generally, and they have a very large intersection. The orange rectangles represent another family, 5S. Its target structure is a Y shape. They also have a very large intersection. But the intersection, so within a family, it is easy to find a set of parameters that give you, that work well across the whole family. But it's basically impossible to find a set of branching parameters that work well between the two families, because not only are this white rectangle and white trapezoid far from each other. The intersection between tRNA and 5S is this little white blip here, but they're also far from the standard parameters. And so what we're currently working on, we've had a recent paper submitted, is the idea that we're going to improve prediction accuracy by actually being able to do it on a per sequence way. We have a new provably efficient algorithm where we can partition the parameter space. And so now we are wrestling with the problem of we can generate a large set of possible structures. And based on our existing data analysis, we can show that there is almost certainly a needle in that haystack, meaning a much better prediction than what you would get under the standard parameters. We're just not sure. We haven't built a metal detector yet. We haven't figured out exactly how to find our needle in the haystack, but it's a part of our ongoing research project. And with that, I'll conclude and thank you all for your time.
Okay, Christine, thank you for a very interesting talk. So the floor is now open for questions. So Christine, I've got a question.
Yep.
How difficult is it to actually discover experimentally the structure, the secondary structure in RNA?
Ah,
Well,
it's
I
a
mean,
great question.
I have
That,
no idea how to do that. I mean, and
yeah,
you mentioned some papers, especially when you had a virus that they think changed its secondary structure in a number of ways to
yes.
for the virus wanted to do something probably bad, so it did that. But
Yep.
how do they know that those are the structures, or those are
Uh, the,
they guess?
yes. So in that paper, that often what they will do, so what they will do is they will predict possible structures. So biologists do very much consider the MFE, predictions. You can consider structures that are sort of uh, suboptimal structures that are close to the MFE, but not added. Often, to test this experimentally, they will do point mutations. So if they think that if they think position I is paired with position J, this is a C and this is a G, then they can mutate. They can change the G to an A, and they will see if that disrupts the functionality. And then in order to show that that was actually what was going on, they can then change the C to a U, so restoring the base pairing, and that should restore the functionality. There's also something called chemical footprinting, and that is a way of probing the structures to measure how flexible they are. And with the idea being that if a position is not flexible, then it's often, a base paired position should be not flexible. But the correlations are not quite as strong as the biologists would like, and so making sense of that data is a problem that I'm actually also working on, and is quite challenging because of the noise in the systems.
So when you say functionality, would they check that by they make the stuff into a protein, then look how does the protein
Ah,
work? Is
so
that what they do?
yeah, so with the viruses, I don't know in particular. I'm most familiar with back at the beginning, I showed you this picture of the Petri dish that had the RNA structures in them. Those are actually RNA that's involved in what is called the quorum sensing system for Vibrio cholera. So Vibrio cholera is the bacteria that causes the disease cholera, and that disease turns virulent when there's enough bacteria. So the bacteria are communicating with each other by like sending out small molecules and taking them up. And those RNA are involved in the regulatory system of determining, like, is there enough of us now so that we'll sort of go on a rampage? And they were, and so they were able to I guess, track in the laboratory the effect of point mutations on the RNA structure, and so determine exactly which position, exactly which positions were critical in terms of the the virulence of the bacteria.
Can you echo, say, point mutations in your own computations?
Ah,
Base?
I have a graduate student who's supposed to be thinking about this is what exactly is the effect of point mutations? Because sometimes they do nothing and sometimes they completely disrupt the secondary structure. And it's not, again, the MFE predictions are not the best way of determining that. And so her challenge is to come up with a better way of assessing the effect of point mutations on the secondary structures.
That'd be very interesting since they can do that experimentally. So that's
Yes, exactly.
yeah.
So there's a potential, there's the Rich Carthew, who's a biologist at Northwestern, who was actually the director of their NSF Simons Math Biosis Center, has a really nice system that he's worked with. And so we're hoping there's a potential collaboration that will come out of that. Exactly, because it is most directly testable experimentally.
Excellent.
Thank you.
How about some other questions, please? The floor is open. Okay, well, if there are more questions, we'd like to thank Christine for an excellent
Great.
talk. Thank you.
Thank you. Thank you so much. Myself.